And rather appropriately, he's from Glasgow, so it's great to have a Glasgow speaker as our first talk. And it's Professor Adrian Bowman from the University of Glasgow, where he has spent much of his career and is now head of the School of Mathematics and Statistics. Adrian's very active in research, currently in environmental modelling and in the analysis of anatomical shapes. I think it's the latter bit you're going to talk to us about today. Adrian's also been very, very active in the RSS. So I'm going to read all this out just to highlight all the things he's done over the years. Joint editor of Applied Statistics, honorary secretary of the research section, member of council, chair of the Careers Promotion Committee, honorary officer for membership, and Guy lecturer, to name a few. So that's fantastic. So can you please put your hands together and give a big warm welcome to Adrian.
Thank you very much, Scott. That's very kind. I've just been around for longer than more people, that's all. But good morning. Welcome to Glasgow as a Glasgow resident, and I hope you enjoy the conference as much as I am. Looking forward to enjoying it. Now, I'm very aware that giving a talk like this is a real privilege because you get twice as long to speak as anybody else in the conference. So I feel a real weight of responsibility in that to make good use of the time. So what I'd like to do is tell you about an area I've worked in with others for quite some time. It's an area I find throws up a lot of really fascinating problems, and it also connects with some really interesting areas of science. And in a way, actually, that captures the essence, I think, of why being a statistician is such fun and why it's potentially a very influential thing to be. So I hope to celebrate that a little as we go along. Now, the best rule in life, if you're looking for career advice, is work with people who are smarter than you, and I've had the benefit of doing that for a number of years, and these are some of the current people, particularly Liberty Wittert and Stanislav Katina, been close collaborators over a few years now, and other people are working on different aspects of the things I'll talk about. There's also been, in the backdrop, a Wellcome Trust-funded project, which is called Face 3D, and that's brought in a wider interdisciplinary group. So, of course, I am absolutely not working on my own in all of this stuff. So, what's the data we've got? Here is a picture of one of my colleagues, Liberty, and it looks an unexceptional picture until you realize that it's actually in 3D. And that's a very commonly available style of photograph nowadays. And in fact, we've got a camera system that I'll show you in a minute, which produces it. You can go and buy them if you have enough money. And what it produces is 100,000 points, which just sit in three-dimensional space. And so it gives you a picture of the surface of whatever it is that you point the camera at. Here's the camera. And if you look on the right-hand side, you'll see two of them there. There's two on the left as well. So if you look at me with your two eyes, if you look first with your left and then with your right, you will see slightly different images. And your brain is fantastically clever at using the mismatch in those two images to work out how far away I am. Which is why it's not a good idea to drive a car if you've only got one eye, because you can't see where the opposition is. But that's the principle of stereo photogrammetry. And of course, there are other ways in which you can do it through laser scanning and so on. Now, most of the time, I'm going to talk about faces, but of course, there's many other things whose shape is of interest. I saw our president tucking into some of these last night. They are very good in Scotland, mussels. And there is a project recently been going on in environmental science in Glasgow, which was looking at a designed experiment to see the effects of climate change. If you grow in a warmer environment and with a higher CO2 environment, what effect does it have? Including on the shape of the shell, which then it's important that has a good closure when required. So that's an environmental issue. There's another project which we've been involved in, which is about breast surgery. I'm not going to show you the pictures, but if you imagine a muscle and you amplify it a bit and a little bit of imagination, you're halfway there. But breast cancer is something which is unfortunately not so uncommon. And if you have breast surgery, then if you have one breast affected and one breast unaffected, there's an opportunity to try to perform that surgery in a way which has the best possible outcome. Again, it's the shape which is going to be critical in doing that. Here's another picture, which is a face of sorts. It's a mouse, and there are many mice or mouse models which are very useful in genetic studies. And we will, at some point during the talk, be looking at some genetic aspects, and mouse models are particularly important. So it's still a relatively recognizable and relatively simple shape. Here's another one, which is a jaw from a tiny little fish. And this is an evolutionary study and environmental study. What's the effect of different environments on the way that the jaw forms in order to scrape off from the stones the material that's of interest? This is a very complex shape. Very large amount of detail with the way the bones fit together being of interest and the shape of the overall object being of interest. So the agenda is, how do we approach analyzing that kind of data? And of course, there's a very long, illustrious history of doing this, going back to Darcy Thompson in Dundee and before that, which it gives an initial platform to work from. But the kind of data, as ever, that we get nowadays is much richer, much more voluminous. So here's the agenda. What I'd like to do is talk about how to build a model, at least in the special case of a face, where we know a great deal. Because what we've got is an unstructured data set. It's just 100,000 points in 3D. We don't have a meaning attached to those points, and so we need a model which will give a suitable structure. As statisticians, we always think carefully about the variability, so let's spend a little bit of time thinking about how to do that. And then there's a couple of areas I'd like to explore, one genetic and neurodevelopmental, the other surgical, and some material at the end, if we have time, just to point forward to where things might go. Now, the starting point, the traditional starting point, because it was all one could do some decades ago, is landmarks. And landmarks are points which have an anatomical meaning that, especially with a bit of training, one might carefully locate on an object. It's laborious, so if we've got hundreds or thousands of these images, this is not going to be a good way to go. But having got the landmarks and put in the work on that, what is generally done is we go for some kind of description of the rest of the object we're looking at. So we go from the points out to a surface and use that surface description in some way to analyze what's going on. Well, I think one of our main contributions to this debate at the moment is it might be useful to think of an intermediate structure, which is a set of curves. And those curves seek to follow the ridges and the valleys of the face of the object, and therefore they capture a lot of the structure by doing that, and the rest of the surface is relatively simple. So there is an opportunity to explore a model for the face which has got this hierarchical structure. The points that are commonly used and understood, the facial surface, which is a full description, and an intermediate object, which is a set of curves. Now, there's not much technical material in this talk, but I should, for maybe towards the beginning, for a few minutes, give you a sketch of what's going on in terms of constructing a model like this. And so let me begin with a hierarchical model, the landmarks L, the curves C, the mesh M, that we can build up in that way if we wish. The data we observe is the image sitting there in 3 dimensions, which might be 100,000 points. And it's going to be natural to think about some kind of Bayesian model here, because of course we know what a face looks like. If I tell you here are the eyes and there is the nose, you know where the mouth should be, and so there's a huge amount of information that we can immediately exploit in doing that. So here is a way of breaking down that posterior, if you like, into the mesh conditioned on other things, the curve conditioned on other things, and the landmarks. We needn't do it in that order, but it would be natural to do so. So let's just look at 1 or 2 components of that, and I've written down the last term in its usual kind of reverse form. And I'm just going to pick out 2 things. One is, where do we get a prior from, for say landmarks? And then the other thing is, where do we get the likelihood from? And I'm just going to pick on the component there, which is the curves given the likelihood. Well, let's think about landmarks for a moment or two. There is a whole body of work which very elegantly synthesizes a lot of the thinking about statistical shape analysis. Look at the book by Ian Dryden Cantimardia, second edition, pulls it all together in a very elegant way. And one of the key things is that when you take images, of course, they don't relate to one another. They're in different parts of space. And so we need some way of bringing those together to be in a common coordinate system, a common space. And registration is sometimes the name given to that in different medical image settings. So if we have landmarks, two sets of landmarks, we can simply move them together because they correspond. Each one has a corresponding meaning. And we use least squares, basically, to fit them together as closely as we can. And then the remaining variation tells us about what's going on. So that's been done here. I've just got a face behind this to show you where they lie on the face. But these landmarks come from different people so that they're now registered, and the variation that remains is the variation that we can study. So that means, of course, that we can maybe write down a multivariate normal distribution, and I'm showing you the different components of here in the Smarties that are embedded in this face. And that is a prior distribution. It's an informative, highly informative prior distribution because we've done the hard work of doing it in lots of individuals. We think we've got it more or less right, and that generates a prior. Well, what about the other component? What about the likelihood bit, if you like? So suppose we've got some landmarks and we're interested in a curve, which we definitely are. How do we go about constructing it? Well, if you're dealing with surfaces, and you've done a little bit of differential geometry, which I haven't, but I've looked up the books, then the very elementary thing that you need to know is that at any position on a surface, we have a description of the shape of that, and it's based on two principal curvatures and two principal directions. That's as much as you need to know about differential geometry, and it's just about all I know about differential geometry. And so if we run through, and it's really a bit like Taylor's theorem. Taylor's theorem is one of the most useful theorems around because it helps you get out of trouble when you can't write down the very detailed analytic work. What we're really saying is that there's a quadratic shape, if you go down to a small enough neighborhood, going on at every point on this continuous surface, and the parameters of that are which direction has strongest curvature, which orthogonal direction has weakest curvature, and the sizes of the curvatures go with those. And so, of course, people have studied that for hundreds of years. And there is a relatively recent suggestion of one of the ways in which you can put all that together into what's called the shape index. And the shape index is just a way of normalizing these curvatures, combining these curvatures, to tell you what kind of surface you've got. And you can see it running through in the video there. You've got a cup, or a saddle point, or a cap, and that's a very useful typology of what you've got at a particular point in a surface. So suppose we take a face. Remember, these are just points, 100,000 points in 3D. And at each particular location, we just take a neighborhood, we fit a quadratic, or maybe better a cubic, and we go off and work out the curvatures. That's a bit laborious, but not too hard to do. I'm laborious in the sense the computer has to work very hard. And so we can just color it, type it, by the sorts of surface shape that are present across the surface. And you can see the red bulb of the nose, you can see the pits of the eyes, the green bits, color-coded that way, the corners of the mouth, and the ridges and the valleys, and blues and yellows, and so on. So that's quite a useful way of beginning to see where some of the features are. And if we look at the landmarks traditionally defined, then they often align with some of those curvature features. Actually, traditionally, they don't. They're about sort of careful positioning and looking at things from the side and working how far forward things are and so on. So one of the things we have been keen to do is to promote not only the use of curves as a richer description in this area, but also to redefine landmarks in terms of the geometry of what's going on rather than the traditional anatomical positioning. Okay, so we've got some information to get us going, some information which tells us what sort of shape the surface has at any particular position. Let's just home in on something simple. Here's a set of closed lips. Remember, at this stage, conditioning on the landmarks, and here we've got two landmarks that tell us where the corners of the mouth are. How do we find the curve that runs across the valley where the closed lips lie? Now, this is dead easy to looking at it. You can just draw a line on it. The point is, of course, that if we've got many images, we want to do this automatically. We want to have some way of tracking that curve and many other curves across the face. How do we do it? Well, first thing is, we're looking for a certain kind of signal in the surface shape. We're looking for this valley signal, the blue bit. And we need some sort of way of tracking around on this. And I suppose if we were being mathematical about it, it's a manifold. It's something that's really two-dimensional, sitting in three-dimensional space. So how do we track around this without actually leaving the surface in ways which will enable us to estimate things? Well, I suppose we always start with something linear, and then we improve it if we need to. Well, here's one way of thinking about how to make this first go linear. We're trying to find a curve, and a curve is just a path across this object. So what we can do is take a plane, and the plane contains the two landmarks that we know are the endpoints of this, and then just allow the plane to rotate. And as it does so, it cuts through the surface. Geodesy, that's what it means. You cut through the surface of the Earth, the surface of this shape. And as we do, what we'll do is try and pick up as much valley curvature as we can. And if you think about that, we just position it there in the middle image, we can see that it's trying to capture where the curvature is of the right valley type. Everything else is kind of zeroed out to blue. And if we do that, then it's the beginnings of a reasonable attempt to say where the curve is, but it's probably not going to be an ideal description of it. How do we define a valley anyway? The valley is not defined in any axis system, it's just sitting there. How do you characterize the points which lie along the floor of the valley? Well, functional data analysis has helped us a lot, hasn't it, in recent years to think about some of these issues. And if we think of a path which is indexed by where does X go, where does Y go, where does Z go, so that's the P at the top there, then we've got this curve defined by P, and we're really looking for a curve where the curvature is maximum. The curvature across the valley at the bottom is going to be maximum. And so we can think about writing down an integral across this curve where the integration is of the maximum curvature, and we want that to be as big as possible. So that's what we've done in a very rough way, just by having a straight line kind of thinking, except it's not a straight line, it's an intersection of a plane and a surface. But it's really in 2D, and so we're probably going to get into trouble if we keep thinking in 3D all the time. So what we need is a coordinate system which will bring us into 2D, because a surface is really 2D, at least locally. So yellow points are the landmarks, black points are the first attempt to find a path here. And what we've got is two coordinate systems, or two axes. One is, for each red point, we simply say, well, where would you project onto your reference line, this linear reference line that we've constructed? Well, we'll project onto the green point, so perpendicular, but it's across the surface. And then that green point has a position along the path, the arc length of the path. So what we've got is an axis system, a new space, and that space is horizontally, how far along the reference path do we go to find the projection, and how far up do we go to find the point on the surface? And so that gives us a more familiar space to work in. We can do all sorts of things there, bring all sorts of tools to bear on that. What we're really trying to find is the ridge where this maximum curvature is maximized. So a lot of technical detail sitting behind it. One more slide just to show that there are very familiar things going on here. The problem is to maximize the integral along this path. And those of you who work in splines in any form, B-splines, P-splines, all that kind of stuff, are a real help to do that computationally. And Newton's method is a way of optimizing it, which is really very efficient. So that's as much of the technicalities I'm going to give you. And if you want to know more, ask me at the end or later. Very happy to talk about it. But I'd much rather talk about the concepts and then why it might be useful to have such a model. So one final step, that we've defined a ridge, but actually it's still in that 2D space. So what we can do is transform it back to the 3D space, and there's something called barycentric coordinates, which help us do that. Let's not dwell in the detail. So the end result is a curve at last, something that you could see by your eyes very easily at the beginning, but now we've got an algorithm that will help us identify that. And it's using these signals of curvature, and in many ways what I'm talking about today is all about bending and stretching, which goes on expressed in curvatures. So we can extend that out across other curves, ridges rather than valleys, and there may be other types of curve that we wish to identify. But what it gives is this hierarchy that I mentioned before, landmarks, which sometimes are done manually, but we're trying to get away from that. Then the curves, which are an intermediate stage, but actually mop up a lot of the information, and they're a much simpler object lying in a simpler dimension, as it were. And then a full surface mesh, if we wish it, which fills in the gaps that the curves otherwise give us anchors for. And so for any particular image, we can write down a model. Now, of course, people do that in many ways, not at all suggesting this is the first time anybody's done that, but it's the particular perspective of having this hierarchy of information that I think we are finding very useful, and it gives you an end result here in something which we can use fit to any facial object and then run off and analyze, which is the bit we really want to get to. Okay, so a model built on, ultimately, that we have kind of Bayesian mechanism to do that. We're not quite there yet, but a model which is using estimation to come up with the things of interest to us. What about variability? Because, look around the room, there's huge variability in facial shape that we see every day. Actually, faces are really interesting things to us because we spend so long looking at them. And most of your day will be spent looking at other faces. I sometimes bus or the train find myself looking at somebody and thinking about research, and you sometimes get odd looks back when you there we are. That's the hazards of doing this kind of stuff. But variability is the key to understanding what's going on in any situation where we want a statistical model. And what we've got here is just an initial and very standard thing to do in this area, which is principal components. One view of what we've got is that a model for a face, although we've got 100,000 points to begin with, we've reduced it down to maybe 1,000. That's still a very high-dimensional object, and leaving aside the kind of formal framework of how we might deal with that, think of it as simply a long vector where each point has its x, y, z coordinate, and you've got a huge multivariate vector. So things like singular value decomposition, principal components, is going to be really useful to help us work in a space which captures a lot of the variation of that, but gets away from the huge dimensionality that we otherwise struggle with. And so in principal component space, we're dealing with the model as a multivariate object, but each point in that space corresponds to a shape back in 3D if we then unpack that long multivariate vector into its corresponding shape. So if we simply take all our faces, register them in the way that I talked about before, and we do a principal components on them, when we go down a particular component direction, first principal component, we can actually turn that into a shape change, because as you move along that component axis, move through that space, each point in that space corresponds to a shape. So the first principal component is that. And it's really a large-scale expression of a narrowing or widening of the face, and perhaps a stretching in other directions, because these things are all standardized by size, by the way. I should have said that. We're not so much interested in how big things are, but what the relative expression of shape is. So that's the first component, which explains quite a lot of variability. And the second component is a little different, and it's something to do, I think, with the way the nose and the mouth separate out and protrude. You get a bit seasick after a while. So I'm going to try and restrain myself from showing these videos too often, but let me show you the first three. And here's the third one, which is something to do with the jaw and a kind of smile, the corresponding effect on the lips, the kind of smile and frown on that. So one can have endless hours of amusement looking at faces, which describes some of the variability in these individual components that we see in this high multivariate space. Now, what I did there was take a template, take an image, and just fit it on top of the model so that you saw a real face. Here is an example of the model. Actually, it's missing the brow ridge. Sorry about that. I forgot to fix that. But there is something that goes across the brow above the eyes. And rather than look at individual components, of course, we never see one component at work. We see many components at work when you look at a particular face. So what we could do, there's something called the grand tour. If anybody's old enough, as old as me, might remember that when multivariate methods were being studied some decades ago. And the idea was you simply randomly track through, you walk through this multivariate space, and you see what kind of shapes emerge. So I'm going to do that. This is just taking principal components as a multivariate normal, randomly sampling, and then tracking between the points. And so what that shows is the kind of combined effect of the main principal components to express facial shape. Now, of course, none of these are real people. These are just artificially generated. And I suppose we should really put a face on it, like I did before. And there's a long tradition of people allowing themselves to be part of their own experiments. And against my better judgment, I was asked when we were going to do a study of reliability, how accurate is this process, this measurement process? Against my better judgment, I was persuaded to be one of the participants. And so there we go. They told me the headband was just to keep the hair out the way, but I think it was just to increase the level of humiliation involved. And if you look very closely, you'll notice there's a couple of black marks, which they said they wanted to measure reproducible points on. So there's one between the eyes and one underneath the nose. There we are. I'm sorry about that, but that's it. So let's get rid of the bits that don't matter for this purpose and just look at the face. And now because we've got a model for that face, and we've got a model which is moving around and showing variability, just going to attach my face to that and show all sorts of... I find this rather creepy because I'm looking at myself wearing the faces of different people. It's really interesting to see the extent to which, because you can see me and some of you know me, you actually recognize the person from the texture and perhaps the wry smile or whatever it is I've got there, rather than the actual shape. But now, of course, we're really interested in the shape here. Okay. Well, we're going to tackle some particular investigations involving shape before too long, but just at this stage of exploring some of the tools involved, what about sexual dimorphism, which just means that males and females tend to look different? Here's a random sample of two of them, and you can see they look different. But, of course, there are so many cues there about clothing and hairstyle and all the rest of it that we have no difficulty really usually, although there's quite a number of cases where that may not be the case. There's usually strong cues about whether it's male or female. But if we do this business of fitting models, and here is a picture of a mean, which is just kind of averaging the registered objects, of a male and female, and the blue here is the male and the magenta is the female, and they're transparent, so they're overlaid. So what we see is that the males tend to have a more prominent mid-face, females tend to have more prominent cheeks, and there are some differences in the brow as well. And if we just using very straightforward standard methods at the moment, principal components, there's no reason principal components should show differences in the first component. That's just about general size and shape variability. It's not until you go much further down the list that you begin to pick up components that really seem to differentiate here. And not completely, of course, but this component does. And if we look at the shape that's expressed on that component, then you can see again it's to do with the cheek and the prominence of the midface and so on. And that's generally well understood. We're just seeing it here expressed in this particular model, and maybe a little bit more detail because it's a full surface model. Okay, so a lot of the time we're going to be interested in means, in the differences between populations and groups. But that's not the only problem that's of interest, of course, in life. And there are issues, and particularly medical, we'll look at some of them, where we've got a single individual, and we want to ask how the single individual relates to, say, a control population. And there'll be cases of that we'll look at. So I'm just going to use the current data to introduce some ways of thinking about that. So imagine principal component space. We've got all these points sitting there, and each point actually corresponds to a shape now. Only draw it in two really, but kind of mimicking three here. Say imagine that as the controls. And what we'll do is to make that, let's say, the females, and then I'm the male. How do I relate to female shape? That's an interesting question. So there's the points that we observe, and we might reasonably fit some kind of multivariate normal distribution to that. And then here am I, or somebody, sitting out here, and identifiably different in the sense that that confidence ellipsoid, or actually prediction ellipsoid, describes the variation that's present in this reference group. And here am I, and I'm outside it. So one interesting way of proceeding is to say, well, what makes that red point essentially, what makes it different from the control group? Well, what we might do is just project it towards the mean of the reference group and ask, well, where does it actually hit this prediction ellipsoid, this normal range, if you like to call it that? And that therefore says, well, that's as close as we can get to you within the reference group. And so the difference between those two shapes might be helpful, might be informative in saying what are the characteristics of your shape that make you different from the reference shape. And that's potentially quite a useful thing to do. And actually doing it is really simple, some very simple equations, and those are the last equations you'll see, I think, but just to indicate that it's not particularly hard to do that. So here's another randomly selected male, although you can probably recognize who he is. What would it take to make me consistent with female? You know, gender nowadays is a topic that's hotly debated and discussed. So what would, in terms of facial shape alone, using the reference group of those who are clearly identifying themselves as females, what would it take to turn that shape into something that's consistent with the female reference shape? Well, there it is. So if I need to be disguised, and I want to be disguised as a woman, here's what I need to do. And you know, my nose is too big for this purpose. The mouth is a little prominent, and it's that cheek area again, which, as we saw earlier, was the standard characteristic difference between the male and the female common shape. Okay, so that was largely a bit of fun, just taking some data that is easy to understand, I hope, because we all kind of relate to that. But what I'd like to do is, for a few minutes, talk about some issues which are genetic and neurodevelopmental, but use the tools to have a look at them. And this is principally work with John Waddington, who's the Royal College of Surgeons and neuroscientist in Ireland, and he has worked in this kind of area for a long time. And I find it really quite fascinating. And when he first told me the origins of this, I just find it difficult to believe, but there we are, it seems to be true. There is, as the fetus develops in the womb, there is a single group of cells which later differentiates into the face and the brain. And you can see that is natural, I suppose, just because of the spatial proximity of those two anatomical things. But it means that if there is some kind of event or some kind of condition which affects that development process, that it can express itself both in the brain and in the face. And so that suggests that the face potentially carries some information about the brain. And that's really uncomfortable, because it relates back to some ancient, notorious, outrageous stuff about how you could diagnose criminals by the shape of their cranium and all that sort of stuff. So it's nothing to do with that. And it becomes a little more plausible when you think about Down syndrome, for example. I have a friend who has Down syndrome, and she has a characteristic facial shape, which I can recognize others who have that similar condition. Now, this is absolutely not about diagnosis. It's about, from the neuroscientist point of view, it's trying to understand the potential developmental origins of some of these things. And Down syndrome is not the only one. Fetal alcohol syndrome is another much studied one. And there's this one called velocardiofacial syndrome. And it's a genetic condition. I think it's understood which gene might be implicated in this. And it affects the brain in various ways. It is thought to affect the face. And indeed, there's links with heart conditions. So it's, I think, a really fascinating area. So there isn't time to develop a really detailed analysis of that because this experiment was quite complex. It was paired data, so there was a case and a control within the same family to try to get some genetic matching. Unfortunately, that sometimes the case and control were of different sex or different age, so that all had to be struggled with. But as I've indicated earlier, principal components are one of the things, I mean, it's absolutely not the only thing to do here, but it's just one of the accessible things that people often do, and so we followed suit in this and ended up doing a permutation test. Because there's an element of design in this experiment, we can protect ourselves against too much assumption and the methods of analysis here, and look at permutation methods, which identified that particular component 2, just put all the cases in the same space. Where is the differentiating? And clearly linear discriminant analysis is another kind of obvious thing to do here. And so that particular component, it made extreme by going out to several standard deviations here. It's not suggesting that these are anything like individuals, just showing the nature of the shape change that seemed to be related to the differences between the cases, velocardiofacial syndrome, and the controls who are unaffected. And I mentioned principal components as a standard thing, but there are other things we might do. And as we go along, I'm just going to throw out a few lines to different directions of development that might work. And this is really my colleague Stanislav Katina, who's working in this particular area. And it's about the fact that you can decompose the variation into what's called affine. That just means it's straight lines, it's linear, including a sort of shear, where you're not allowed to have local deformations which bend. It's always straight. And so it's big scale stuff. And correspondingly non-affine, which you can localize in various ways and look for features which you know are going to be local rather than just look across the whole object. And so there are ways of doing that which are intended to home in rather than looking at a kind of shotgun approach of principal components, which actually often is very effective, I have to say, but it can look at particular features which the neuroscientists can then go off and try and understand what those shapes, how they relate to other studies that have been done. And we're not in a position here of coming up with a blinding revelation. It's putting the jigsaw pieces together over a period of time to try to understand the origins of this. And actually, there's a study being analysed at the moment, which is schizophrenia and bipolar disorder, and there is a hot debate, I understand, about whether that is has neurodevelopmental origins or not. And so we've got data which is controls, schizophrenia patients, and bipolar patients, and this particular component at the moment seems to be emerging as something which suggests that there is a slight difference in facial shape on average in that manner across these groups. And so that's the subject of current investigation. So, shape, genetics, evolution, and so on, we'll come back to that at the end if time permits. What I'd like to do for a moment or two is talk about a completely different setting, still medical, still faces, but surgery, and work with surgeons in Glasgow, principally Ashraf Ayub, and Bowman Kambi is now in Birmingham. And they're involved in dealing with the faces of people where often there needs to be some kind of surgical intervention. And one of them is called orthographic surgery, and it involves moving the jaw. And you'll see two patients here where the jaw is in a position where the teeth don't meet, and sometimes, as teenagers as well, facial shape's very important, and their psychological implications and so on. So sometimes people go in for orthographic surgery. What does that involve? Well, this is actually a CT scan of some patients before and after, and you can see the enormous difference that has made to the facial appearance by doing that. And what goes on is that underneath here, there's the CT scan, we can look underneath, is the bone, and this surgery involves moving the bone, either the mandible, the lower part, or the maxilla, the upper part, in order to bring them into closer alignment. And as you see in the after image, there are evidence of what's done. I think actually breaking the bone and putting something in or taking something out or whatever. It's very clever surgery. And it does have a big impact on facial shape. So, does it work? How do you answer that question? Well, one answer is that you get some experts together, you look at the before, you look at the after, and you say, yes, that's good. Another approach is to quantify it, and that's the role of a facial model. Then there's a way of expressing the shape change, and in particular, the post-orthographic surgery. How does that relate to control shape? And what is control shape anyway? That's a kind of difficult question to answer. So what we're doing here is limiting it down to a particular group of people who are defined in census terms, as it were, as white UK or something, you know, the way that the census breaks these things down, because of course ethnic origin, we'll come back to that, has potentially a big effect on shape and so on. So we need a homogeneous group in order to do this. Having got that, we've then got a group of controls, we've got a group of post-orthographic surgery patients, and we're interested in, well, are there systematic differences between them? Has the surgery just completely moved this back into control shape or not? Here are, again, you know, one would do this more globally. I'm just showing you the components, 1 to 10, and a device which I find really useful is when you're looking at lots of stuff, use color to show the ones which are well separated. So there's a scale here which is basically a t-statistic, and it picks out a principal component 3 by color as the one where things are potentially a little different. And this is maxilla Lafort surgery, as it's called, a particular way of operating, and we can see that on average, the reds, the post-surgical patients, the jaw's still a little bit further forward than it normally would be. And there may be good reasons, of course, why you can't just arbitrarily move things into any old place. But it's quite a useful audit tool to say, on average, this is the outcome. What's that telling us? So there's discussions can go on about what that means. Then there's also this idea of closest control, because we're dealing with an individual patient. Dealing with the populations, very important question, but dealing with an individual patient is also important. So that idea of closest control, the reference group, the control group, after surgery, is that individual, how do they relate to that group? And so projecting on, in the way I described earlier, is one approach to that. And this is pre-surgery, so we see it's the gray is the control, and the purple, the magenta, is showing us the ways in which the individual differs from the closest control that we could find to match that shape. And it's not surprising that pre-surgery there'd be big differences. For this particular patient, fewer differences after surgery, but still some. And of course, one might not expect that, as I said earlier, that this is going to immediately take you right into the control region, but it has improved. It has moved some areas and not others, and again, there can be a discussion of what that means for the patient. Here's another one, similarly, where the whole of the lower jaw, which is the most important part here, is really very close to the control group, and there's some kind of transfer up to the cheeks, which might be considered. So shape as a whole, but there's one particular feature of shape which is of interest, which is asymmetry. And something is very asymmetric, we generally pick that up visually quite clearly. If it's a small effect, probably not. But if there's strong asymmetry, then we're very good, I think, visually at picking that up. So if there's a model sitting behind here, and the model itself is symmetric, so the bits on the left correspond to the bits on the right, and then what we can do is measure asymmetry by the obvious device, taking the model, flipping it, and then just relabeling left as right, matching it up, and any remaining deviation is asymmetry, due to asymmetry. Because if the thing is perfectly symmetric, whatever the thing is, it's going to match exactly when we take its mirror image. So that's good. We've got a measure of asymmetry. Here's a patient. It's actually a post-orthognatic surgery patient, but that's not the photograph of the patient, as you are. One advantage of having the model is we can attach a template, which is somebody's face. I know whose face it is, but I'm not saying. Which can then, because of the model, just be mapped on. And so we don't need to show the identity of individuals, which is useful. Is that face asymmetric? Well, I suppose all faces are asymmetric. Mine certainly is. But in what way is it asymmetric? Can you see, give you a moment or two to look at it, and how would you characterize where the asymmetry is and what it is? And is it big? Is it small? Well, if we've got the model and its reflected image, reflected version, we've matched them together, then just point by point, we can say, how far away is my partner? And do that over the whole image. And we might say, how far away are you in the X direction, or the Y direction, or the Z direction? We could split it out if we want to, and there's other kind of ways we could express that. And of course, showing 3D movement is quite tricky. So what I've done in the next slide is just separate out the mismatch between the original and its reflection, and then coded that, or used color to code on the original image whether you move a lot or a little. So it's topographic colors. Green means you don't move very much. Blue means you move in one direction, purple strongly, and then the brown means you move in the other direction. So that separates out X, Y, and Z. And actually, there is a bit of asymmetry around the nose, and there's a bit of asymmetry up here beside the eyes. Maybe some of you spotted that. But if we do this device of just showing the animation, which sometimes helps, what that's doing is going from the original image and it's saying, how do we get to complete symmetry? That's what we do. We just tilt it that way a little bit. And in any face that we look at, there's bound to be some degree of that. But at least we've got a way of measuring it. So, orthognathic surgery. Here's a control. And so if you measure asymmetry, color coding it, there's very little in that control. But pre-orthognathic surgery, there may be a lot of asymmetry. And that's something that it would be useful, potentially, to adjust as the surgery takes place. And what you don't want to do is to introduce asymmetry, because that may have unwanted effects. So, got a method now of measuring that. And so if we go to a particular patient, a kind of score sheet, which I need to explain for a moment. First of all, we've got on the right the images pre and post. You can see the big difference in shape. On the left is a kind of scorecard. And I don't know if you're familiar with density strips, but I'm a big fan. Chris Jackson came up with this suggestion a long time ago. It's just a way of showing a distribution using the density of ink. So the grey-black bits are the control population. That's what sort of asymmetry score you get in a control population. Now we can just mark on the card, as it were, where that patient is before and after, and quantify the extent to which things like the lower face asymmetry, missing the LO, which is second from the bottom, that's, of course, the one we're most interested in, because that's where the surgery is going on, and you can see a very strong degree of improvement. Okay, what I'm going to do is, because time is pressing, I'll skate on a little bit and tantalizingly show you a few slides I'm not going to talk about. But the cue there is that there is another line out into different ways of analyzing this, because I've been talking about fairly straightforward methods. And if you've got curves, which I've been trying to promote as a really useful part of the building this model as a whole, what about analyzing the curves? And of course, there's a lot of interest in that. And in particular, there's interest about separating out the phase change in a curve from the amplitude change. There's a very good paper in statistical science discussing those kind of issues. So there's a whole set of issues there. What I'd like to do is to reassure the chairman is finish in the next couple of minutes and talk about some stuff that's now going on, and it's longitudinal. So we've now got these images, we've also got the images over time. And the time scale can be very, very different. So here's a picture of somebody smiling. Maybe you want to see that again, if you happen to blink at the wrong moment. But that's quite interesting to see how people smile quantitatively. But it's also particularly interesting if you're doing surgery, cleft lip and palate, which is about the mouth. It's sometimes when people smile that the shape differences are exhibited. So there's a whole set of issues there and how you model that longitudinally. We've also got a really interesting set of data based on earlier work, which looks at a small-ish group of children from 3 months now to 15 years old. And we have their three-dimensional shape at various time points, not as many as we would like, but at various time points along the way. So my colleague, Liberty Vittert, has helped in constructing this image, which is taking a single individual, 3 months, and letting that evolve on continuous time scale to grow to age 15. And so you immediately see the baby features disappearing and the adult face beginning to emerge. And if we do that in profile, you'd see the same thing, the very chubby baby face very quickly stretches. Remember, the time scale here is uniform, and over 15 years. And then the adult face begins to emerge with the features. I find that really very interesting to look at. What we need is a model for all of that, a longitudinal model, which we've just about got. I'm just showing you one individual case, but the fact we've got a model for each image is what enables that to happen. Well, I have one more little issue to discuss, which is nose shape, and this is with my colleague Vincent Macaulay and Adrian Marinas, a student who's just been finishing on this, which is back to the genetics and evolution. It will take about 1 minute max to do this. So, if we take African, Chinese, and British people, then it's another surprising fact that, genetically, you can use current measurements of genetic structure to track back to find out how things may have evolved over time. And you can do that with, apparently, with movement of peoples and all the rest of it. It's fascinating stuff. So underneath that is a tree, a phylogenetic tree, which assumes that there was a common ancestor. And what we're interested in is the ways in which that may have bifurcated out to create a descent pattern that now gives us what we see in A, B, and C. And if you write down a Gaussian process model is the standard sort of way of approaching that in genetics. And now what we'd like to do is to attach a shape to it, not think about the genetic structure, but ask how has the shape changed over time? And this is the tree which emerges, which is showing the Britain on its own. There's an unusual feature, isn't it? And the African and Chinese faces having a common ancestor before the global common ancestor, if you like. And so the final picture really is just to show you what the model describes for these curves as an expression of nose shape, and the three pale blue curves are the three populations, British, Chinese, African, and British noses are the ones that stick out further and are a little bit narrower. And that is, in purple, the best estimate of the common ancestor of African and Chinese noses, whereas this one, on the right, the red one, is the common ancestor of everyone, and you can still see that it's much closer to Chinese and African than it is to British. Well, there we are. I've had a tour of all sorts of different areas of science. Underneath it is this issue of shape. I apologize for using up all the time allotted. Happy at the chairman's discretion to take some questions. Happy to linger or speak to you later in the conference, but thank you very much for listening so patiently.